Right. Did you do the questions?
Yeah.
Right. So, did you do that question paper which I sent you, word one?
Have a little bit in that, but I did the checkpoint on fully.
Right. You did checkpoint question. I didn't. No.
Yeah, the checkpoint on the second one, the up and up and up and up. So,
Okay. Right. So, can you please read out the answer?
um, motor neurons are structured like to efficiently transmit signals from the CNS or central nervous system to the muscles or glands by contracting. And if we take first the cell body, which we call the soma, it contains the nucleus, which is like the center of, which is like in the center of the neuron. So it synthesizes proteins needed for the neuron function, receiving incoming signals from other parts. And the dendrites receive signals from the other neurons and transmit them towards the cell body. And if you take the axon, it is like a single long projection that carries signals away from the cell body to the muscles. And the myelin shell is like a fatty insulation layer that surrounds the axon, which speeds up the signal transmission by jumping between the gaps called the nodes and the axon
Nodes
terminals,
of
which is
Ranvier, right? Nodes of Ranvier.
nodes of Ranvier, and the axon terminals, like at the end of the axon, releases those neurotransmitters that helps to stimulate the muscle fibers at the neuromuscular junctions. And the neuromuscular junction is like a specialized synphase, which is like where in the motor neuron, the axon terminal like meets the muscle fiber, which allows the transmission of the signal, which causes in the end which in the end causes the muscle to contract.
Yes, very good, right? So that is the answer. How it is, how your structure of the motor neuron is related to its function. How the structure is going to relate to its function, right? Okay, so then we'll start the next one. That is like nerve impulse. So what do you understand by nerve impulse? What is actually a nerve impulse?
It's like a electrical signal.
Yes, it is a signal, right? It is an electrical signal. So how this is going to generate by the charge differences across the neurons membrane. By the charge difference mean what?
It's like the ion charges or like
Ions, yes, ions. Ion charge difference. So in that case, it is going to create this charge difference and thereby creating the electrical signals, right? It is just like a potential. You can tell it also as an action potential, right? We'll come into this action potential and all later. Actually, this is a change in the electrical charge. So thereby it can cause the signal. It can cause an electrical signal, right? So how this electrical charge is created? It is mainly across the neurons membrane, right? So it is allowing this communication between the neurons and also neurons, muscles, and the glands as well, right? So it arises from the movement of the ions through specialized protein channels and is maintained by active ion pumps. So there are active ion pumps. So by that case, it is going to create this charge difference of the ions, right? So this process is going to facilitate the transmission of the signals. So when you are if you have asked like what is a nerve impulse, so you have to tell it as an electrical signal and how this electrical signal is generated. That is also important. So you have to know, right? and then myelinated nerve fibers. So we have discussed about these myelin sheaths, Schwann cells, nodes of Ranvier. We have discussed little bit, right? So in vertebrates, most neurons are covered by the Schwann cells. So most of the neurons in the vertebrates are covered by this. Why they have given here mostly invertebrates? Yes, they have given here like vertebrates. So can't you find this in invertebrates?
Yeah,
Huh? Yes or no?
with N, right?
No, invertebrates, they are not having the myelin sheath, right? Only the vertebrates is having, right?
Okay.
Why is that? Because this myelin sheath actually is like a insulating material, insulating layer. So we talk about this, right? So this is producing by the Schwann cells, right? The Schwann cells mainly in the peripheral nervous system. And then the nerve impulse is going to jump between the nodes of Ranvier. So this is going to increase the conduction speed. But what about these invertebrates? So I told you that they are not having the myelin, right? But they don't have myelin, but they are having the axons, thick axon. So that thick axon, it can increase the conduction speed. That is enough for the invertebrates, right? So some of these invertebrates, they are going to show the features of this myelin sheath, but true myelin sheaths you cannot find in invertebrates. Understood? Right. So you have to know that vertebrates, they are going to have this myelin sheath, right? So wrapping around the nerve fibers. You know why? What is the importance of the myelin sheath? Why there is a myelin sheath? You know, right? We have discussed. So this sheath, it is going to protect the nerve and it is going to speed up the impulse transmission. Gaps, there are gaps between these Schwann cells. It is known as the nodes of Ranvier. So it aids in the process of nerve impulse transmission. Why? Because the nerve impulse is going to jump from one nodes of Ranvier to the other node of Ranvier. So thereby it is increasing the speed of the nerve transmission along the neuron. Right? So that is the most important thing here. So they have given one diagram also in your book. You can just see it if this one is not clear. Right? This is the Schwann cell they have given, and this is the part of the axon, right? How this Schwann cell is going to wrap around the axon, they have shown here, right? I think these words are not clear, so you can see it in your book. Now here there are three Schwann cells, and the space or the gap is the node of Ranvier. Right? And in this Schwann cells, you can find the nucleus as well, right? So this is the myelin sheath. Right, and then the speed of the nerve impulses. Nerve cells transmit impulses rapidly, right? And it is influenced by the fiber's diameter and the presence of the myelin sheath. Now, if you consider about the nerve impulse transmission, speed, right? The speed of the nerve impulse. So, what are the factors which is going to affect for the speed of nerve impulse? Yes. What are the factors affect for the nerve impulse? The speed of nerve impulse?
No, the question again, I had a signal drop.
Yes, I asked what are the factors which is effect for the nerve impulse speed or speed of the nerve impulse. Factors affecting for the speed of nerve impulse.
It's like it depends on like the diameter of the myelin cells diameter.
Yes, diameter. What else?
Then we take like temperature, like some higher
Temperature.
temperature,
Right? Temperature.
the
Okay, how this temperature is going to affect the nerve impulse transmission?
like the rate of chemical reactions increase and the ion movements also get fast.
Right. That means you are telling that temperature is going to increase the impulse speed. Right. In
Yeah,
cold climate, then what happens for the impulse transmission? In cold climate,
it will be slow.
slow. Why is that? Why it is going to slow? Right.
Is it
Now
like
ask. Yes. Yes.
is it like some kind of like if the like temperature drops the pressure in the nerves also may
Pressure of the nerves. Pressure of the nerves in which pressure you are talking about?
like after that happens like pain signals will be delayed and still it slows down the transmission of like the impulses like that.
Uh, Actually, this low temperatures, it is going to slow down the nerve impulse speed. That is correct. Now here in lower temperature, what can you tell about the kinetic energy of these ions? It will be slow, right? The kinetic energy will be slow. So in that case, it is going to go for the less transmission. And then enzyme activities also will be reduced in low temperatures. So the proteins will get denatured. So it is going to affect the nerve impulse transmission. So likewise, the low temperatures, it is going to affect the nerve impulse transmission. That is correct. Temperature is going to affect. Other one is what you told. What is the other factor you told? One is temperature, other one is diameter. Now here, when you are telling about the diameter, you have to tell which diameter. Now here also they have given fibers diameter. Which diameter here they're talking about?
Axon, no,
Axon. Yes, axon. They are talking about the axon, right? And then diameter of the axon is also matters for the speed of transmission. And what else? What about this presence of the myelin sheath? So myelin sheath has to be present. Why? Because it is going for this impulse which can transmit by jumping between the nodes of Ranvier. So this conduction is known as the saltatory conduction, right? That means the impulse can be jumped from one node to another node. So thereby myelin sheath is also one factor which is going to speed up the nerve impulse transmission, right? Nodes of Ranvier. That thing also you can take, right? It is also very much important in nerve impulse transmission because the nerve is going to jump from one node to another node, saltatory conduction, right? Okay, so those are the main factors which is going to affect. Now, fiber's diameter and the presence of the myelin sheath, they have uh, talk about those two factors in this sentence, right? Thicker fibers and myelinated nerves carry impulses faster. So you know that myelinated nerves, it is going to carry the impulses faster. And here, thicker fibers. That means the fiber's diameter. They're talking about the axon's diameter. If it is thick, so thereby it can increase the nerve impulse transmission, right? The speed. Vertebrates, they're having these myelinated fibers, rapid responses up to 120, while invertebrates, they are relying on the giant axons for the fast impulses. Now, if you consider about the uh, vertebrates and invertebrates, as invertebrates do not have these axons, but still they are having the thicker axons. So if they are having the thicker axons, that means their impulse transmission also can be speed up by that, right? But in vertebrates, they are having more complex than that. They are having the complex component, that is the myelin sheath. So their impulse transmission is somewhat more than this one, right? Higher than that. And then we have to investigate the nerve impulses. Now, am I audible to you?
Yeah, man.
Right. So, investigating the nerve impulse. So, how you are going to investigate this nerve impulses? Early studies used external electrodes to measure the electrical changes, but were limited by mixed nerve signals. Hodgkin and Huxley advanced research by using microelectrodes to study the giant axons of the speed, enabling precise recordings of nerve impulses. And then modern techniques now allow detailed studies of individual nerve fibers. Now here they are talking about the investigating of nerve impulses, right? So these scientists, they have done various experiments to find out this nerve impulse transmission. So they make the experiments mainly with the electrodes. You know what are electrodes?
Anodes and cathodes.
Huh?
Electrodes means like the positive and the negative side.
I didn't get what you told.
It's like the electricity changes in the signals, like positive to negative, like something like.
Yes, actually, these are like the materials which are used to detect or you can measure the electrical activities, right? So thereby you call it as electrodes. So there are the different methods that you can use these electrodes. You can use different materials for the electrodes, right? So now here they have, what is the experiment that they have given here? This one, right? Electrodes. Okay, they didn't given much more techniques about this investigation of the nerve impulses, but I will tell you something about this one. They have actually, the scientists have used different methods to investigate this nerve impulses, right? That means what they are going to investigate here, they are going to investigate how the electrical signals are going to generate, how they are going to transmit, right? And is this transmission is affected by the external factors. So those are the main things that they have to research on, right? So they have used different methods. So they have one method is they have given here like the squid. Who is a squid? You know. Now here they have given a one experiment they have used these microelectrodes on squid. Do you know who is a squid?
Yes, ma'am.
Yes, you know, right? So here, their axon is somewhat large as it's an invertebrate. So what they have done is they have inserted these micropipettes, microelectrodes to their axon, right? And after that, what they did was they experimented on this by putting or by inserting the electrodes, right? Because if the axon is large, you can insert the electrodes easily. So they have inserted some microelectrodes to their axons, and they discovered about this role of ion movements, about the role of this sodium plus ions, potassium ions, how their charge difference is having, right? And then what are the potentials, right? And what are the responses that they have got? So all this they have experimented using these giant axons of squid. So that was one experiment performed by this Hodgkin and Huxley, right? And they have used different electrodes also in finding out these nerve impulses, right? They have got some electrodes, micropipettes, so they have measured the voltages of the axons, right? So these voltages were helped in identifying the potentials, electrical potential across the nerve membranes, right? Across the membranes. So those are the different experiments. So this is one picture that they have given here in your book. So I didn't get the what is the explanation here. Just turn the page number. Uh, just wait. Page number 239.
Okay.
Right now here, this is the figure D. Right now, what is this? This is an apparatus where they have used internal and external electrodes. Right. So this the internal electrode you can find right into the axon. They have used one electron, and outside also they are having a part right of this electrode. So it is used to examine how this neuron is going to work, right? And they have found out the potentials of the neuron. They have measured it, right? The resting potential and all potential differences across the membrane. Why across the membrane? Now here you have learn about this cell membrane structure in AS, right? So they have given here the cell membrane, right? The membrane, neuronal membrane, right? So here axon membrane. So here you can get the electrode. Part of that is inside, intracellular, right? Other part is outside, extracellular electrodes. So here they have measured the charge difference by these electrodes, right? And they have used here in this picture you can find it, right? Two electrodes, one inside and one outside. So it can detect the charge difference of the voltages. Voltage difference they can measure. And they have got it to a graph like this. Right? So what is this? They have found out there is a constant difference of minus 70 millivolts between outside and inside the resting potential. In resting potential, this is the voltage. Right? By doing this experiment, they have found out this minus 70. Right? That is the voltage in resting potential. What is resting potential? Any idea about resting potential?
It's like an interval.
Huh? Resting mean what?
It's like an interval.
It is like what? I didn't hear.
It's like an interval.
Yes, actually it is a charge difference, because here you are going to measure the charge difference, right? Outside and inside. So if the neuronal membrane is not going to send any nerve impulse, so this is the voltage, right? That means this is like a starting point of a nerve impulse. It is not going to send the impulses. It is at resting stage. That is the resting potential. They have found out the resting potential value as minus 70 millivolts. This is the value of the resting potential, right? This is a typical value. Understood? Any questions? So we'll see about this resting potential and all. I think you have to study later chapters. Okay, here you have to learn about the resting potential. Right, how the nervous system is going to work. Right, how the nervous system works. Nerve impulses. You know what are nerve impulses. There is an ion gradient. Change in charge difference. Right, that means a charge difference. That is a gradient. So sodium ions and the potassium ions are distributed unevenly across the axon membrane. Now, if you consider about the axon membrane, you can get the sodium plus potassium ions uneven distribution, right, across this axon membrane. And then membrane permeability. Sodium plus is relatively impermeable. Potassium plus is freely permeable. What is this? What it says here? Now, actually, you have to know what is the role of the sodium plus and potassium in nerve impulse, right? So, this sodium and potassium is very much important. The ions are very much important for the generation of this nerve impulse and also the transmission, right? It is important. So, their movement, now they can move, right? So, by their movement, they can generate the changes in the voltages. So thereby it is going to produce the signals, right? Action potentials. Now, if you consider about the right now here action potentials, different thing they have given, right? Now here they have given like membrane permeability, right? What is this membrane permeability mean? You have learned about the permeability in UAS, right? I guess.
Yes, sir.
Yes. Now membrane permeability mean what?
It's like the ability of a substance to go through like
Yes,
biological membrane.
right. So that means how the ions are going to cross the membrane, right? How easily the ions can cross the membrane. So that is the permeability. Now here they have given membrane permeability. Sodium plus is relatively impermeable. That means what? When compared to the potassium, it is somewhat impermeable. That means their transmission is somewhat slower than potassium, right? Their crossing is slow than the potassium. But potassium is freely permeable. It can move across the membrane, axon's membrane. So that is the permeability, right? How easily these ions can cross the membrane, right? Okay, so then what is this resting potential? Now we have talked about resting potential when the nerve impulse is not transmission. It is at resting stage. That is the resting potential. It is minus 70. We have found out, right, using the experiment. So maintained by sodium potassium pump. Pumps 3 sodium ion out and 2 potassium ion into the axon using ATP. Potassium is diffuses out via the open channels, leaving inside slightly negative. So the potential difference is coming like this. Inside negative relative to the outside. Membrane status is polarized. Right, now how this resting potential is maintained? Now they have given here about the resting potential, right? You know the voltage of the resting potential, that is minus 70 millivolts, right? You know what is this resting potential? It is the when the neuron is not transmitting, that is the electrical charge difference, right, across the membrane. That is the electrical charge across the membrane when the neuron membrane is not going to transmit the impulse. So that is at rest. So we talk about the resting potential, right? Now how this resting potential is maintained? So you have to know about the sodium potassium pump. How it is going to maintain? Now this pump, this is an active transmission, right? Why? Because it is a pump. Pumping mean what? It is going to pump using energy, right? So this is an active transportation against the concentration gradient. Why there is concentration gradient? Because there is actually a gradient of ions, right? So the amount of ion, the concentration of the ion is different. So there is a gradient. So they are going to transmit or transport these ions against the gradient. So they are going to transmit it using energy. So it is an active transportation. Understood that much?
Yes, sir.
Right. When the three sodium ions are going to pump out of the cell, what happened? 2 potassium will come into the axon, right? That means 3 potassium ions are going out of the membrane, 2 potassium is going, 2 potassium is coming in. So thereby it is going to create high concentration of sodium. Why? Because that is 3. So the concentration of sodium is more, right? Now, where can you find the high concentration of sodium? Is it in outside or is it in inside? Yes, outside, right? Because why? 3, 3 sodium. So more ions are in outside,
Okay.
right? High concentration of sodium ions is in outside, and potassium high concentration is in inside or outside? It is inside. Sodium concentration is high in outside, potassium concentration is high in inside. So there is what then? Inside the neuron, you can get negative charge, right? Inside the neuron, you can get negative charge. Did you understand that one? Now here, that's why here, potassium diffuses outside via the open channels, leaving the inside slightly negative, right? Because more sodium is in outside and
Ma'am,
potassium
can you
is
repeat
in.
the sodium inside?
Huh? But what you asked, I didn't get. Did you tell anything? Am I audible to you? Am I audible now?
Yeah.
Right. No, we have stopped here like this. The 3 sodium is going out and 2 potassium is going in, coming in. So then you can get inside the axon more negative. Why it is more negative inside the axon?
Potassium diffuses out.
No, potassium is coming in, right? Sodium, more sodium is going to go out. That means more positive charge is going out. More positive charge is going out means less negativity is there inside, right? Because more positive is going out. So inside there is negative. So that's why it is saying that leaving the inside slightly negative. So inside is negative, right? Inside negative relative to the outside, it is negative. So therefore the potential difference is minus 70 millivolts, right? Polarized mean what? Membrane status. Now they are telling about the membrane status. No membrane. That is polarized. That means polarized mean, now here they are talking about the membrane, right? Axon membrane. Or then polarized mean inside it is negative compared to the outside. Outside of the neuron is positive. So that means it is unequal distribution of the ions. So in that case, we call it as polarized, right? Because inside it is negative, outside it is positive. So there is a unequal distribution of the ions. So the membrane is polarized. Membrane status, that is polarized. Understood this one? What is resting potential? How it is creating? Now you have to know how the resting potential is going to generate. So this thing you need to know. Sodium potassium pump, right? How the sodium is going out, how how this potassium is coming in, how the inside is creating as negatively charged, and how this membrane is going to polarize. So there is a charge difference. So that is resting potential. Any questions about this?
No, ma'am. Understood.
Right. They have given the diagram also, right? The resting potential. Now they have given here the resting potential of the axon is maintained by the sodium potassium pump. So you have to know, that is very important, how this resting potential is maintained in the axon. It is by the sodium potassium pump. You know how much sodium is going out, how much potassium is coming in, right? The relative permeability of the membrane and the movement of the potassium ions along the concentration gradient and electrochemical gradient. So there is actually a gradients, right? Electrical gradient, ion gradient, or mainly the concentration gradient of these ions. Now here in this one, sodium potassium pump create concentration gradient across the membrane. Here you can see sodium is going out, potassium is coming in to the axon, right? This is the inside of the axon, right? Yellow one. So here potassium is going out, sodium is, sorry, potassium is, no, sodium is going out, potassium is coming in, right? Low potassium ion concentration outside. Outside what you can find? Outside more sodium ion concentration. And so potassium ion concentration is less in outside. Why? Because potassium is going to come in now. So outside you can get more sodium compared to inside, right? And then these are the channels, right? Ion channels. So by this, potassium is diffusing out of the cell down the potassium concentration gradient, making outside of the membrane positive and the inside negative. Now here inside is negative. Why? Because more positive ions is going out, right? So the electrical gradient will pull potassium back into the cell. So there is a gradient now. When you consider about the potassium, there is a charge difference. So they can pull the potassium again back into the axon, right? So here at minus 70 millivolts, potential difference, the two gradient concentrated counteract each other and there is no net movement of potassium. So here at 70 millivolts, what happened? It is at resting potential. Resting potential means the two gradients will be counteract each other and then there will be no any net movement of the potassium. So then you called it as a resting potential at minus 70 degree, minus 70 millivolts, right? So this is the diagram they have given in your book. You can just see it, right, in your spare time, right? And then we have to learn about the action potential. So before going into the action potential, did you understand this one? How this potassium thing here, potassium channels, right? So if there is any questions, you can ask, right? I can explain it. Right. We'll do one question, please. Okay, can you just do the third question of page number 239?
Okay.
Finish writing.
This
Yes or no?
did it plan, but not sure can we.
Okay, what is the answer you wrote? Did you write anything as the answer?
Yeah, I wrote, ma'am, vertebrates achieve like a faster transmission speeds despite having a small diameter of fibers compared to some invertebrates due to the presence of the myelin sheaths and the literary conduct. And I also wrote the nodes of Renwiers in the myelin sheath is not continuous, but it and efficiency I could polarize the membrane. Like that's all I wrote.
Yeah, so that is it, right? Now here when you are talking about the question says like this, right? Speed of transmission of nerve impulse is in part related to the diameter of the nerve fiber. Vertebrate nerve fibers always have relatively small diameters, vertebrates, but the speed of transmission is high. So they need the explanation. How? Why do these vertebrates they are having the faster nerve impulse, right? Even though their nerve fibers are smaller than the invertebrates, right? Because of this myelin sheath, right? You wrote about the myelin sheath. So vertebrates neurons are wrapped with this myelin. So this myelin is helpful in the saltatory conduction, as I have explained. That means nerve impulse is going to jump from one node of Ranvier to another. So thereby it can increase the nerve impulse transmission, right? So they don't need a large axon for this transmission. Large diameter they don't need. Invertebrates actually they are not going to have the myelin, but their conduction is also somewhat rapid. Why? Because they are having larger diameter axons, right? So, however, if you are considering about these large diameters of invertebrates, their conduction is somewhat slower than the saltatory conduction of the vertebrates, right? Understood? So that is the answer,
Understood.
right? So then we'll go for this action potential. Now here you know that when a nerve impulse is going to travel through a neuron, the following steps can be occurred. Right? First, there should be a stimulus. What is this stimulus? Stimulus means you know what is this stimulus, right? Stimulus mean actually an what is actually a stimulus? Just tell me. Do you have any idea about the stimulus? What is it? So I don't need any
It like
definition.
triggers the neuron.
What?
It like triggers the neuron.
Actually, this is like a change, right? So it can activate your sensory receptors. By any stimulus, you can activate the sensory receptors. Now here a response. Stimulus like light, sound, touch. So all these stimuli, they can activate your receptors, right? So the axon membrane becomes more permeable to the sodium ions. Now here, for an action potential to be generated, there should be a stimulus always, right? There should be a stimulus. So when you are going to talk about the action potential, so there should be always a stimulus in order to generate the action potential. So that thing you have to remember, right? The word depolarization. What is this depolarization? Now membrane is polarized during resting potential. So that part you know, right? Depolarization means your sodium ions channels open, right? Now here in the resting potential, these are closed. But in depolarization, what happens? Sodium ion with the stimulus, when it is getting any stimulus, it is going to open up the sodium channels. That is depolarization. And when it is going to open the sodium ion channels, what happens? Sodium is going to coming in or out. It is going to rush into the axon, right? When the sodium channels are open in the membrane, the sodium ions will come in. So it causes the axon to become positively charged. Now, in the resting potential, it is negatively charged. But when the sodium ions channels open and sodium will coming in, so it will be positively charged compared to the outside. So it is something like plus 40 millivolts. So this sudden change in charge is called the action potential, right? This sudden change in the charge by opening up of the sodium plus ions, it is known as what? Action potential. And what is the time where it lasts? It is at about 1 millisecond. Right here in this diagram, you can find it out. Here, this is the resting potential. When you are getting any stimulus, what will happen is your sodium plus channels will open. Then sodium from outside is coming in to the axon. So thereby it increases the positive charge of the axon inside. So then it is now we say that is depolarized, and then it is going to reach to the threshold. That is the action potential, right? And then repolarization. Repolarization means what? In repolarization, what is happening is your sodium ion channels close, right? Sodium ion channels will close and potassium ion channels open up. When potassium ion channels will open up, what happens? So it allows the potassium ions to move out of the axon. Now the potassium ions that is there will move out of the axon. This is happening because of the concentration gradient, right? So potassium will move from inside, it is going to move out, because inside you can get more potassium, right, compared to the out. So here potassium will going out. That is repolarization, right? So then what happened when the positivity is going to reduce now? Why? Because the potassium ions is going out. So the axon inside of the axon, it will become negatively charged compared to the outside. Here, this is repolarization, right? You can see here how it is coming, right? After reaching here, repolarization starts. Repolarization, sodium plus ions will close, potassium is going to open up, and potassium from inside is going out. Now inside it will become more negative, right? Like this, it is coming. And then what happens? Here they didn't mention about this hyperpolarization, right? Hyperpolarization mean actually this potassium channels are going to close slowly, right? These potassium ion channels are going to close slowly. So in that case, what happened? Sodium ions is going to move out, right? So the negativity become more than this minus 70. The negativity becomes more than the minus 70. That's why it is coming down from this line, minus 70. Right, later on what happened? Here you can get again the restoration. How this restoration is taking place? Sodium, potassium, ion pump is going to restore the ion again, ion positions. So the membrane, it reaches or it returns to minus 70 millivolts again, right? So repolarization, you know, and then you can add hyperpolarization also if you want, right? Hyperpolarization means so potassium ion channel is going to close slowly. That means more potassium is going out. So it creates more negativity inside. So that's why the graph, the line comes down, this line, minus 70. Right, and after that, again the resting potential. Resting potential means restoration of the resting potential. That means what? Here again the sodium potassium pumps is going to actively restore the original ion balance. Now they are going to restore the balance, right? Restore the ion. So then the axon is coming to the resting state again. So this state, right? And it will come to the position like this, right? Sodium channels is coming in, sorry, going out. What is it? Yes, sodium is going out and 3 potassium is going out, right? So we talk about this one. And then here the leakage from the leak channels, again the potassium is coming in. So thereby it can restore the ion balance, right? It comes to the resting potential again. Understood how this action potential, depolarization, repolarization, hyperpolarization is happening? Any questions?
No, ma'am. Understood.
Right, and then all or nothing response. What is this all or nothing response? This is also very important. So you have to know that action potential is going to occur only. Action potential is generated only if there is a stimulus, right? If that stimulus also should be very strong to open the sodium channels, right? That stimulus should be strong enough to open the sodium channels. So thereby depolarization is going to the threshold level, right? Once triggered, the size of the action potential is always remaining the same, right? So that means this one, right? Now here you can get this thing. Now here you just getting, now here minus 70, it is resting potential. A stimulus is getting, but that stimulus is weak. So then what happens? Anyway, you are getting a stimulus. Then what happens? Your sodium ion channels will open, but few, right? Few of the sodium gates is going to open. So thereby it is going to go for the depolarization, but it is not going to the threshold, right? Threshold is something like plus 40. So it is not going to the threshold. So then this is a weak stimulus, right? So can you see an action potential here in the first graph? Can you see an action potential in the first graph?
There's no action potential.
No action potential, right? Now here in the second one, can you see an action potential?
Yeah, ma'am.
Yes. Why is there an action potential in the second one? Because there is a strong stimulus. So more sodium pumps will open, right? More sodium pumps will open and there is depolarization, and depolarization is reaching to the threshold level. That means it is plus 40 millivolts. Now it is remaining at this point, action potential. There is, right? And then it is coming to the repolarization and hyperpolarization. So strong stimulus, many sodium gates will open. So depolarization is reaching to the threshold and action potential is generated. Right? Now here also, if there is a very strong stimulus, there also an action potential is generated, and action potential is not, it will not be a larger size. It is saying like that in a very strong stimulus. Right now, can you tell me why the in a very strong stimulus the action potential size is not large? That means why there is not a large action potential? Can you just think and tell me? Did you understand my question? I'm asking in a very strong stimulus, why the action potential size is not large?
Depolarization.
That means,
The threshold is depolarized and it creates a current.
yes, actually, in a very strong stimulus, uh, just wait, right? Right. What was the question? It's like why the size of this action potential is remaining same, right? When you are applying a very strong stimulus, still the action potential size is same. It is not going to generate a larger size. So I'm asking why, right? Actually, so we have discussed about all or nothing response here, right? What is this all or nothing response? That means actually a nerve impulse is going to happen either fully or it is not going to happen, right? That means action potential. Action potential is going to happen fully or either if it is not going to happen at all. That means what? Now just think that the stimulus is going to reach up to a very strong level, but action potential is going to trigger, but it is remaining only at the same size and the shape. That means minus 40 at sorry plus 40 at this level only, right? It is not going to go beyond this. So that is because of the all or nothing law, all or nothing principle. Yes, understood. It is always remain at that point, at that position. So you have to remember, very strong stimulus, it is not going to increase the size. That means the amplitude of this action potential, right? Understood?
Yes, sir.
Right. So these are the diagrams, right? You just go through these diagrams. It is very important, right? You have to know the graph also. Sometimes in the examination, they will give this graph also to identify what is the resting potential, and you have to know that is minus 70, and what is the action potential, that is plus 40. You have to know, and what is this one, depolarization, repolarization, and if it is going beyond this minus 70, that is hyperpolarization, right? And so these things you have to know, and the shapes also. So when you are studying, you go with these diagrams always. It will be easy for you to understand, right? And you have done this AS examination, right, before? Yes. So you got the results of that?
Yeah, ma'am. I have to do unit 3 again because I didn't have time to do it.
But
I had to do unit 3 again, the practical ones, because I didn't have time to study for that because of the crash.
okay, right. Now, how you're getting these results? Now you face the examination in unit 1 and unit 2. Is that?
Yeah, and
Yes. So
it
you
it
got the
usually
results of that?
yeah, I had 1A and 1B for that.
Okay, good. So why you are not telling me the results then?
I told man, I told a
You
long
told
time ago.
me.
Yeah,
Okay, I
it
can't
came on
remember.
March and you asked and I told you. Man.
Okay, I didn't remember whether, because I was wondering whether you got the results or not still, right? Okay, now you have to do the unit 3. So how was it? You have that part, right, with you?
I have the PDF, man.
Yes, you can do it. And this one you have to complete in October,
Yeah, man. And after that, it's over.
right? Okay. And then we have to know, we have to go for the refractory periods. And after that, we end. Let's do some questions. And okay, we'll do this one also, and after that, we'll do the questions. Refractory period. What is this refractory period? That is given like absolute refractory period. Sodium plus channels completely blocked. Impossible to stimulate the membrane. Relative refractory period. Stronger than normal stimulus required. Potassium channel still open. Resting potential not fully restored. Now, in the refractory period, you can have two things, right? One is absolute, other one is relative refractory period. So, in absolute refractory period, what happens? It is the time period is also not given here, right? But it is actually about 1 millisecond. Now, here in the absolute refractory period, you will not get a second action potential, right? That means what? No matter the stimulus is strong or not, you will not get any other action potential for the second time. So this type of absolute refractory period is happening in the depolarization state, right? So why it is happening in the refractory period, absolute refractory period? Because the sodium channels are going to blocked. That means inactivated. So then they will not going to open again. They cannot reopen again. So that's why another action potential cannot be generated for the second time. Understood? Because the sodium plus channels are blocked. So that is absolute refractory period. So did you understand what is this refractory period? The word?
Yes, ma'am.
Yes. Now actually refractory period mean it is the time where one action potential has occurred, right? And it is the time where, or else you can tell like this, it is the time after the action potential, right? Now just think one action potential has occurred and then the time after this action potential. So that is the refractory period. right? So in that period, the neuron cannot get another action potential. This is the reason why sodium channels, they are going to block completely. So they cannot generate another one, right? In this, that is absolute refractory period. Now, relative refractory period mean what? In relative refractory period, here relative refractory period, it occurs after some time, after some time, that means about 1 to 5 milliseconds after the absolute refractory period, this will occur, relative refractory period. Right now, here in this period, second action potential can occur, right? But the stimulus should be stronger than the usual one, right? So this relative refractory period, this will occur just after this hyperpolarization, right? Why this is happening, the relative refractory period? Now, here they have completely blocked just after one action potential, another one cannot do. But after the absolute refractory period, some of these channels will reset, right? But potassium is still leaving, but sodium channels, some have reset, right? So then it can make the second one. So that is the relative refractory period, right? Now you have to know, in absolute refractory period, can you have another impulse in this period, absolute refractory period? Absolute In absolute refractory period, can you have an impulse? Can you generate an impulse?
Is it no?
No, right? But relative refractory period, you can generate, but the stimulus should be stronger than the usual one, right? So that thing you have to know. That is the difference between these two. Right now, this refractory period, relative refractory period is very important. Why? Now, this is going to ensure the unidirectional flow of the impulses, right? So only in one direction the impulses can occur due to this relative refractory period. And also the signals are not going to overlap because of this relative refractory period, right? So you have to know, refractory period means the time after the action potential. Absolute refractory period, all your sodium channels are blocked. So stimulation of the membrane and generating of another impulse is impossible. But in relative refractory period, as it is occurring sometime after the absolute refractory period, some of the sodium channels have reset, right? So therefore, another potential can be generated, stimulus can be generated, but impulse can be generated, but the stimulus should be stronger than the usual, right? Right. Here they have given like key points: all or nothing response. Action potential size is constant if the threshold is reached, right? Now, size always the same. Sorry, always the size is same. Whether it is strong or not, size remains same, right? When it is reached the threshold, if it is reached the threshold, the size is same in the action potential, and then that is due to this all or nothing response. Directionality, impulses flow in one direction, so that is due to the relative refractory period, right? Frequency limit, that is maximum impulse rate, is 500 to 1000 impulses per second. This is the maximum impulse rate. 500 to 1000 impulses per 1 second, right? So this relative refractory period is important as it is not going to overlap the impulses. And also it can maintain the unidirectionality. Right? So understood about this refractory period. Any questions to ask?
No.
Right. Mm, mm. So this is the end of this one, right? This part. Now you can turn to page number 243. 243. Just do the first question. Describe resting potential of a neuron and explain how it is maintained. Did you do this one? No, right? Describe the
No.
resting potential of a neuron and explain how it is maintained. Just do that question first, and after that we'll do the other.
What was the question?
Question, wait. (243). Question number 1. First one. Got it?
Got it.
Right. So do that. you just do the second question also there, right? Okay.
Okay.
Okay, we'll discuss the questions, answers in the next class, right? So we'll stop the class now. Do you have any questions to ask? Any questions to ask?
No, ma'am. Understood.
Okay, then we'll leave, right?
Okay, ma'am. Thank you.